Charles Darwin, when he wrote Origin of the Species in 1859, he knew really very little about genetics and virtually nothing about DNA. The DNA molecule had not been discovered yet. So these concepts that we're going to be talking about during Darwin's lifetime, he would have had no knowledge of. He wouldn't have known anything about it. And so he could not use DNA, he could not use genetics, he could not use the human genome to argue for evolution because he didn't have any knowledge there. Now, as we talked about when we looked at our lesson on the historical aspects, Mendel was around then, and Darwin could have known a little bit of Mendel's work, but Mendel didn't know about DNA either. He knew about genetics, but not DNA. And so, as we said last time we met, homology was the primary evidence that Darwin gave for evolution. He looked at the fossil record. The fossil record was lacking, and he understood that. He thought the fossil record would eventually be shown to prove evolution, which it has not, and we will talk about that in a later class. So what did he look at? He looked at homology, the similarities between organisms. And as we've seen, that doesn't prove DNA, it doesn't prove evolution, it proves a common designer. So about 100 years later, after Darwin, in the early 1950s, 1953, the structure of DNA was discovered by Watson and Crick, famously known. We're going to look at that historically later on tonight as well. Once the structure of DNA was known, then its implications from a hereditary standpoint became to be kind of obvious. It didn't take long. Crick hypothesized the sequence hypothesis. We talk about that in the instructions that I've given you, the book I've given you. He didn't know that for sure. It was later proved to be accurate. His hypothesis was accurate. And so the genetic implications became very obvious. And then finally, a few years after that, 2005, when the Human Genome Project was completed and we started learning about the genome, which is basically another way of just saying the genetic makeup of organisms and the DNA makeup of organisms, then this started, for the evolutionists, this really started to fuel their fire, because what they saw in the human genome and what they saw in DNA and the genetics there, for them gave basically absolute proof that evolution had occurred. I mean, it was kind of a slam dunk, if you will, especially when the human genome was known. We're going to look at all that, but the reasonings for that, Francis Collins, sometimes it gets confusing because you got two Francis's. Francis Crick, the discoverer of the DNA structure, along with James Watson. But Francis Collins was made head of the Human Genome Project by President Clinton years ago. And when the Human Genome Project started. And when they started doing the Human Genome, they expected it to take a long time to figure out the human genome. It actually started going much quicker. They developed ways of looking at the DNA a lot quicker. And so it came out pretty quick, relatively speaking, in 2005, 2003. Francis Collins, who was a medical doctor, but also a geneticist, was the head of that human genome. And it may seem like I may be picking on him a little bit in this class, but he's written a book called The Language of God. It's a very good book. And I read the book, and I've read his arguments, and because he's condensed those arguments into that one book, we will be looking at that. But all evolutionists, to a large extent, hold to what he believes. Now, happens that Francis Collins is also a Christian. He believes in Jesus Christ. He believes in the historicity of the New Testament. But he also still believes in evolution, and we'll talk about that later. So why does he hold to that? Well, he says what lies between the genes plus the findings of similar sequences on the genomes of other distantly related organisms gave powerful support for Darwinian theory. So basically, it goes back a little bit to homology, if you will. You see a similarity in sequence. You see the DNA sequencing, and you'll know what that means when we finish this class. When you see that sequencing, then that gave proof that there was something going on from one species to another. And then what lied between the genes. Most of the chromosomes are not made of just genes, okay? They're made of DNA, and a lot of DNA, but they're not even made of, chromosome is not just DNA either. And the DNA is not just genes. There's a lot of information between the genes that are in the chromosomes. And what this did for Francis Collins, it gave him proof that evolution had to have occurred. Not just Francis Collins, but the other evolutionists as well. Now keep in mind, they didn't need proof. They had already accepted it, but this gave them more and more evidence. For example, if you look at the order of genes along a human and a mouse chromosome, sometimes the sequences are maintained over significant stretches of their DNA. Okay, so if you're, again, kind of like homology, if you're looking at DNA and you're looking at the molecule of DNA and you see that, well, a mouse has a lot of the same DNA that humans do, the only way that you can explain that is evolutionary, right? Well, not right, but that's the way they theorize. And then the other thing, which we mentioned just a second ago, was what's in between the genes. And the genome is riddled with a lot of quote-unquote useless information. For the evolutionists, that useless information was powerful proof for evolution. It was leftover information, left over from the evolutionary pathway that got one organism to another organism. It didn't make sense, and it does not make sense from an evolutionary point of view and from the evolutionist point of view to have a whole lot of useless information in DNA. Why is that quote-unquote useless information there? And so those are the two biggest arguments that Dr. Collins gave in his book, Language of God, The Language of God, to try to show that why he believed that evolution is a fact, and you can't ignore it. It's so much a fact for some of these people that Philip Kitcher called the intelligent, when the human genome was discovered and published, he said that the intelligent design movement was a dead science. This just killed it. This was the thing that killed it, was the human genome and genetics. Some have even called, in your paper I have that, the intelligent design were creationists dressed up in tuxedos, you know, so really making fun of them, not looking at what they had to say. So the question is it true genetics and the genome have given a death blow to creationism? Is that true? And does the intelligent design community have no answers concerning the evidences of evolution in the genome? Well, if you're in this class, you realize they probably do have answers. And we're going to look at that. And they've got some very compelling answers. And I would even go so far as to say, after you see their answers to what Francis Collins and the evolutionists have, that their answers make a whole lot more sense. But if you're reading, if you're a student, or if you're just casually reading the book, The Language of God, like I did, you go, wow, that's just overwhelming. I don't know how you answer that. I could see how that could happen. That's why we're going to spend some time on looking at that book, because those are the concepts that evolutionists hold to. And we don't need to be afraid of that. We could say, oh, I just believe there's a God, and I just, I have the faith that the creation occurred according to the book of Genesis, and I'm just going to totally ignore the science. Well, that's crazy, is it not? That's putting your head in the sand. We don't need to do that. We don't need to do that. Albert Einstein once said, science without religion is lame, and religion without science is blind. Okay? There's no reason to ignore science. Good science. Science that makes sense. Science that follows a logical sequence. There's no reason to ignore it. Even Galileo, who was criticized, as you recall, and even considered a heretic for his belief on the planetary system and denying that the sun revolved around the earth, but it was the other way around, he understood that we were given certain traits by God. We were given intellect by God. We were given these things by our Creator, and I think He intended us to use them. To advance. We're a lot further along now in the 21st century than they were 2,000 years ago. And, I think people live longer now because of that, and medical science has come a long way, so on and so forth. So Galileo said, I do not feel obliged to believe that the same God who endowed us with sense, reason, and intellect has intended us to forego their use. And there have been some good things that have come from scientific discoveries, even scientific discoveries who come from evolutionists. They will find some good things for us to learn from, especially when it comes to genetics and so forth. Not the evolutionary part, but the other things that come from it. You understand what I'm saying? And so we don't need to put our head in the sand and say, well, I have no explanation for that, but I'm just going to believe what I want to believe. No, there's no reason for Christians to deny the science. So, how long have we known about DNA? As I said in the paper I gave you, it's often, Watson and Crick get all the publicity, don't they, when it comes to DNA. Everybody thinks, well, DNA was discovered in 1953 by James Watson and Francis Crick. That's technically not true. They discovered the structure of DNA, but DNA in the chemical compound has been known since as early as 1869. Now, 1869 was only 10 years after Origin of the Species was published. So this guy would have been a contemporary of Darwin. But they didn't know exactly what they had. But he took, kind of a gross thing to talk about, but he went to hospitals there in Germany, or no, I guess it was Switzerland where he's from, and looked at some hospitals that were throwing away bandages on people that were in the hospital for infectious processes. And he took those bandages and he took the pus, if you will, from the bandages, got that out of it, distilled it out, and started looking at the nucleus of the white blood cells that he was able to find from those pus-stained bandages. That's what pus is, it's white blood cells. And he looked at the nucleus and he took that chemical out and he discovered that chemical and he called it nuclein. Now, he didn't know all of the chemistry behind it yet, but he knew that there was something there. The chemical makeup would take a few more years to be discovered, and it wasn't really discovered fully until 1909. So by 1909, you know, not that long after Mischer, that we knew the chemical makeup of DNA. And the interesting thing that was found out about this was that when you looked at DNA, every time you looked at DNA of any organism, whatever that organism was, when you distilled it down, there would always be the same exact amount of adenine and thymine, and there would always be the same amount of guanine and cytosine. Now, adenine and thymine may outnumber guanine and cytosine, but those two chemicals would always be the same together. There would always be equal amounts of adenine and thymine in the DNA that they would look at, and equal amounts of guanine and cytosine. That's an important concept. And you'll see why that's important when we look at the structure a little bit more. But the chemical makeup was known, but the structural makeup was not known. And as I said also in the paper I gave you, there were basically 3 teams worldwide that were really looking at trying to find the structure of DNA, because it became apparent that the key to the functionality of DNA lied in its structure. There was something special about the structure of that molecule that made it real important. So you had Linus Pauling in the United States and his group that were working on this, feverishly working on it. You had Lawrence Bragg and Mac Puritz at Cambridge University in England working on the structure. And then you had Maurice Wilkins and Rosalind Franklin at King's College, also in Great Britain, working on the structure of DNA. And you had other people too, but these were the 3 main factions that were working on it. Rosalind Russell in particular was looking at DNA from looking at it using radio diffraction, and we'll talk about that in a minute. Looking at it from that standpoint, trying to visualize the molecule. But you also had 2 kind of upstarts, James Watson and Francis Crick. They were kind of young rock stars, if you will. Nobody knew them. I mean, they didn't know who these people were. They were at Cambridge, but the people at Cambridge kind of just sloughed them off. Watson had just received his PhD degree at Chicago University. He's 23 years old. That's pretty fast to get your PhD, by the way. But he got his PhD there, looking to work and to do something, and he somehow made his way over to Cambridge and had, I think, an invite to go over there. And then he met up with Francis Crick, who was a physicist, not a biologist. And Francis Crick hadn't even received his PhD yet. Now, why these guys thought they could figure out the structure of DNA, I don't know. But it turns out most people could tell you who James Watson and Francis Crick are. Most people would know Linus Pauling, but because of his work in other areas. He was a Nobel Prize winner himself. But I dare say very few of us would know who Lawrence Bragg or Mac Puritz were. Some people might know Rosalind Franklin only because of what happened with James Watson and Francis Crick. But these other people kind of faded away a little bit. Watson and Crick will forever be known. But they kind of cheated a little bit. I'm not sure how they got a hold of Rosalind Franklin's DNA molecule, the radio diffraction images, but somehow they did. And like I say in the paper you read, contrary to what a lot of people believe, scientists do not share their knowledge when they're trying to discover something. They're kind of vain about that because they're looking to win the prize, aren't they? So I promise you, if Linus Pauling would have had Rosalind Franklin's x-rays, he would have known exactly what he had. There would have been no doubt. But he didn't get them. She didn't give them away. She had befriended, I think, Watson or Crick, one of the two, a little bit, but still it's really unclear how much she really shared or how much they somehow were able to get. You understand what I'm saying? But once they saw this, now I wouldn't be able to tell this was a double helix molecule by looking at that, but that's DNA. That's Rosalind Franklin's pictures of DNA taken by X-ray. And to her, it was pretty obvious. This probably is going to be a double helix, but they didn't know how the double helix was going to be formed. They didn't know how the bases and the phosphates and the sugar molecule were all going to come together. So she even postulated that possibly this was a double helix. But once Watson and Crick got a hold of it, they went to town. Okay, they began to realize what was going on. Now, keep in mind, these other researchers didn't have all that information. Obviously, Rosalind Franklin did, but she hadn't quite put it all together yet. She wasn't 100% sure. When Watson and Crick started getting their cardboard models and everything out together, then it became very obvious to them, at least, what the DNA structure was. And they realized that it was a 5-carbon-based sugar, deoxyribose, in the phosphate group. These were on the outside. They put those molecules on the outside, and they put the nitrogen bases on the inside. And it was those nitrogen bases forming together with the other deoxyribose and phosphate on the outside that formed that double helix molecule. The nitrogen bases were connected to one another in a strand-like fashion in a double helix manner. And we'll look at that a little bit more detail in just a minute, but they realized that. And so they very quickly, I think they realized that these other people were going to be on this pretty quick. So they very quickly published in the Journal of Nature in 1953, great year, the year I was born, I think a few other people who were born that year, molecular structure of nucleic acid, a structure of deoxyribonucleic acid, DNA. And as a result of that, eventually they would get the Nobel Prize. Unfortunately for Rosalind Franklin, she didn't get that prize. She died before the prize was given. Now, would she have gotten it as well with them? I think she probably would have. I don't know. But you don't give the Nobel Prize posthumously. But she was never given the full credit. And certainly Watson and Crick didn't give her all the credit that they should have given her in the discovery of DNA. Their names everybody knows, and not as many people know her. By the way, she died of ovarian cancer at the age of 37. Probably due to all the radiation she was using in her studies. That's been theorized, at least. Who knows, but is unfortunate. But we'll remember in this class. This is the structured DNA. I hope you can see that on the board. Because this, and I know I said we're going to have to do some science tonight, didn't I? So this is real important, though. Because you have the phosphate group represented by a circle on the outside. And then you've got the sugar group, that's the darker color circle on the inside. And then you have the nitrogen bases that are on the very inside of that molecule. And as we will see, they line up always the same with A to T and C to G. And that's extremely important. Now that's what it looks like schematically. If you wanted to look at it in its double helix, it would look something like that, okay? Always A to T, adenine to thymine, and guanine to cytosine. That's the only way they will ever come together. You will never see adenine joining with guanine or cytosine joining with thymine. Now you understand why when you look at the DNA molecule, there's always the same percentage of those chemicals. You will always have the same percentage of A and T and the same percentage of G and C. Now one may be 40% of the DNA and one 60%. They're not always 50-50, but there's always going to be the same of those two molecules because that's the only way they come together. So right at first, Watson and Crick didn't realize exactly what they had. They knew the molecule, they knew it was important, they knew the structure, but I don't think they realized the genetic implications it had. But it didn't take Crick too long to come up with a sequence hypothesis. And after he came up with a sequence hypothesis, and the sequence hypothesis was proven, then it became obvious that now life was not just about matter and energy, but information. And it's the information capacity of DNA that makes it unique. That's it. The molecule itself, we'll talk about this a little bit more. The molecule itself, although a complicated molecule to some extent, and especially the size and the way it winds around and how much of it we have in our body, in and of itself is not the deal. The deal is the information capacity that it has. And this was realized pretty quickly. So you'll see evolutionists even using these terms. They'll talk about genetic information. They'll talk about the message of the genetics, genetic message. They'll talk about the genetic blueprint. They'll talk about the assembly instructions. And they'll talk about a digital code. Well, how do they think that code got there? You know, they use those words, but they say it all came about by chance. But once again, it's information that's important that we want to look at. Now, this is Webster's definition of information. Information is the communication or reception of knowledge or intelligence. That's what information is. You don't get information from dead stuff that don't have the ability to communicate or to understand it if you don't have the ability to receive. And that's obvious. And yet, what seems so obvious to us, or to me at least, that this information could not have possibly come about spontaneously by itself, is not so obvious to the rest of the scientific community, and I don't quite understand that. Even Dr. Collins, who believes in evolution, understood that no current hypothesis comes close to explaining how in the space of a mere 150 million years, that's his years, he's an evolutionist, he believes that the Earth is 5 billion years old, just like most scientists do. But in the space of 150 million years, the prebiotic environment that existed on planet Earth gave rise to life. He understood that. He understands the complication of that, the complexity of that molecule and how difficult it would be for it just to appear. Stephen Meyer says evolutionary theory could not explain the origin of the first life because it could not explain the origin of the genetic information in DNA. Even if you found the origin of DNA, you're still going to have to find out about the information now it contains and who put that information in there, and the sequence of those codes that are in there. And finally, along this line, John Sanford quoted from James Hodge's book, Creation versus Evolution. Its very existence is a mystery, talking about DNA. Information and complexity which surpasses human understanding are programmed into the space, a smaller space than an invisible speck of dust. Mutation, selection could not even begin to explain this. It should be clear that our genome could not have risen spontaneously. The only reasonable alternative to spontaneous genome is a genome which occurred by design. And we'll see why when we go through this study. And like I said, the DNA molecule itself is not really all that special. It's complicated and everything else, but the DNA provides the substance for the information. It is not the information itself. This comes from George Williams, who is an evolutionary biologist. Evolutionary biologists have failed to realize that they work with two more or less incommensurable domains, that of information and that of matter. The gene is a package of information, not an object. The pattern of base pairs in DNA molecules specifies the gene, but the DNA molecule is the medium. It's not the message. Understand where we're coming from? It's what's contained in it, the base pairs. And again, this comes from an evolutionary biologist. This is not a creationist. And Francis Collins, as we said, has called it the language in which God has revealed life. Even our friend Richard Dawkins has said that DNA is uncannily computer-like. And computers have what? They have a programmer, do they not? So, DNA is a polymer, meaning it's more than one molecule. It is a nucleotide. Each one of those base pairs, sugar pairs and phosphate, is called a nucleotide. When you put those nucleotides together, you got polynucleotide. So you'll see those words sometimes. And as we've already seen, it has three basic molecules, the 5-carbon sugar deoxyribose, the phosphate group, and a nitrogen base, guanine, cytosine, thymine, and adenine. DNA is found in chromosomes. I think you know all this. Where are the chromosomes found? They're found in the nucleus, hence the word nuclein when it was first discovered. It's a very unique molecule. It is 19 million times its width. That's a real skinny molecule, okay? And if you were to take DNA from one of your cells and just stand it up, it would be 6 feet tall, more or less, in just one cell. Now, if you were to take all of the DNA from all your cells, which it's estimated that humans have 50 trillion cells, that's a lot of cells, and then you lined up that DNA, it would stretch to the sun 600 times. I think that's amazing. DNA contains a 6 billion letter code. Chromosome 1, for example, has 270 billion base pairs. And if you were to write those base pairs, A, T, C, G, and you just kept writing them over and over and over and over and over again, you would fill up 200,000 pages, and that would be in just one chromosome. What's the point? Well, chromosomes code for the production of proteins from the amino acids. Probably should have a comma after protein, but you form proteins from the sequencing of amino acids. I'm going to briefly discuss how that works, and it will be very brief. And then we're going to see a little video that I found from Dr. Meyer's book, Signature in the Cell. And actually, this video was taken from the movie Expelled that was done years ago by Ben Stein. How many saw that movie? This video is actually taken from there. They got permission from Dr. Meyer to use his book and made the video. But basically, DNA is a double-stranded molecule. It's a double helix, right? Think of this, and you got the base pairs A, T, C, G. You got those that are in the middle. If you were going to try to make a protein, think of a zipper. You'd have to undo the zipper. And so you would have to uncoil the DNA, right? And when you uncoil that DNA, now you've got the individual base pairs exposed. So RNA, messenger RNA, comes in, and it has a similar structure. It only has uracil as one of its base pairs, and it's going to line up based upon that code with the DNA. And 3 base pairs, or codons, make up 1 amino acid. There's 3 base pairs for each of the 20 amino acids that our body has. And proteins are made of amino acids. So depending on the sequence of the amino acid, you will get your specific protein. So the RNA molecule comes in to the nucleus, goes to the chromosome. Now the DNA is unraveled. You've got polymerases and things like that that cause that zipper to get unzipped, if you will. They line up with the sequence, and the sequence is going to be already prearranged. That information is already there by the DNA, right? The ribosome is going to attach to that, and then it's going to break away. And it's going to take that information then to the body of the cell where proteins will be made in the ribosomes. But it's going to base those proteins are going to be based upon those sequencings of the nitrogen bases. So it's fairly simple, but think about how quick all that has to occur in the significant ingenuity behind it. So we're going to look at a little schematic here from Signature in the Cell, and it's going to do really a much better job of explaining it than I just did.
In 1957, Francis Crick first proposed that chemicals called bases along the spine of the DNA molecule function as alphabetic characters in a written language or digital characters in a machine code. This animation shows how this digital information directs protein synthesis. First, a large protein complex separates the tightly wound strands of the DNA to prepare it to be copied. During this process of transcription, a protein complex called a polymerase produces a single-stranded copy of the original instructions. Here we see this copy, a messenger RNA molecule, being constructed inside the polymerase as individual bases are positioned and added to the growing strand. Now we see the polymerase in action from the outside as it spits out the messenger RNA transcript. Next, this RNA transcript approaches and passes through a molecular machine called the nuclear pore complex, an information recognition device that controls the flow of information in and out of the cell's nucleus. Now we see the genetic assembly instructions on the messenger RNA approaching and arriving at a two-part chemical factory called a ribosome, the site of protein synthesis. As the messenger RNA transcript passes through the ribosome, the process of translation begins. During translation, a mechanical assembly line builds a specifically sequenced chain of amino acids in accord with the instructions on the transcript. These amino acids are transported from other parts of the cell by molecules called transfer RNAs, which link specific sequences of bases to corresponding amino acids. The sequential arrangement of the amino acids determines the type of protein constructed. When the construction of the chain is complete, it is transported to a barrel-shaped machine that helps fold it into the precise shape required to perform its function. After the chain is folded into a protein, it is released into the outer cytoplasm to do its job in the cell.
So the key thing on all that is, and for this class, is not to memorize all that and so forth, but to understand that the proteins are determined by the sequence of the amino acids. The amino acids are determined by the sequence of the base pairs, the nucleotide base pairs that are in DNA. It's information, and each protein has a different sequence. Now the average protein has about 150 amino acids in it. Some have more, some have a little bit less. And so there's a lot of different proteins in our body, and each protein requires a different sequence of the amino acids. And again, that's a lot of information going on. The biggest question, the million-dollar question, is how did these digitally encoded, specifically sequenced instructions arise in DNA to begin with? Who wrote the program? Did it just arise spontaneously? Stephen Meyer would say, simple regular geometrics plus rules of chemistry affinity, chemical affinity did not and could not generate the specific complexity present in functional genes. Jacques Monod in 1971 said the code is meaningless unless translated. The modern cell's translating machine code consists of at least 50 macromolecular components which in themselves are coded in DNA. The code cannot be translated otherwise than by the product of the translation. The code had to already be there. It couldn't have established it itself. It would not make any sense. And I may be getting ahead of myself just a little bit, but also keep in mind that proteins, the structure of proteins is amino acids. Now this film sort of demonstrated that a little bit, but these proteins are not arranged linearly. They're folded. And it's actually the fold of the protein that gives it functionality. That's how it works. If the fold isn't proper, the protein doesn't work. So it's more than just DNA sequencing that's going on here. It's the folding that's going on. And so the other question is, which came first, protein or protein synthesis? DNA requires protein, but then it's involved in protein synthesis. So what makes the origin of life and the genetic code a disturbing riddle, I don't know why it's disturbing, not to me, but it's disturbing to some people, is this: the code cannot be translated except by using certain products of its translation. And this is an understatement, I think. This feature of life is exceedingly difficult to imagine, and this comes from British philosopher Sir Karl Popper. So information requires specificity, order, and complexity, not just a bunch of numbers put together. And we're going to show that. We don't have much more time, but I'm going to try to get through some more of these slides before we stop. And I want to show you something that conveys information. Now, if I looked at that, it wouldn't convey any information to me. It's a lot of zeros and ones put in a certain order. Does anybody recognize what's going on here? It's a binary code, right? That's how your computers work. This particular binary code would actually be the very beginning of the Declaration of Independence, if you were to translate it with the information you have to be able to translate it when in the course of human events. That's the binary code, and it's not two base pairs, it's two letters, it's zeros and ones. Here's another code. Now that just looks like a bunch of ACTGs to me, doesn't it you? But when it's lined up in the proper sequence, and this again, not to belabor the point, but when it's lined up in the proper sequence, it produces a specific protein. And this particular protein is RNA polymerase that occurs in a living cell. And every protein in your body has to have these letters lined up perfectly. Does it not line up perfectly sometimes? The answer is yes. Sometimes mistakes are made. Guess what? DNA has the ability to correct its own mistakes. If you want to use the word proofread, it can proofread this and tell when one of those T's and one of those C's or G's is out of sequence. It can proofread it, which is amazing. But when it doesn't, every now and then, something escapes and we get a little bit of an error in the coding mechanism. What would we call that? We call that a mutation. And nobody in this class, I'm sure, understands mutations occur. And creationist and intelligent design community does not deny that mutations occur. We're going to look at that in depth, but we're going to see that mutations could never explain evolution, though, and for various reasons. So the code clearly points to a programmer. Stephen Myers called this the DNA enigma. The enigma is a kind of a mystery or a riddle. The mystery of information needed to build the first organism. Where would that information come from? And Watson and Crick, or Crick especially, realized that it couldn't just show up, and Dr. Collins realized that too. The code, in other words, pointed to a programmer. The complexity of the DNA molecule is not found anywhere else in nature. There's nothing else like it. Now some would say, well, crystals, they can form, spontaneously, but crystals don't have complexity, they don't have information. Also, polymers are similar to DNA, but they lack specificity. So many have tried to determine mathematically how complex molecule could have naturally arisen, and by all accounts, this is an understatement as well, by all accounts, the likelihood is very small. And we're going to see, we have to quit, unfortunately. I thought I'd get through this, but it's a lot of material there. We have to quit now, but we're going to see just how implausible this is next week. And we're going to be using concepts of what I call large numbers. These numbers are inconceivable to us on a normal basis. I mean, you may have a little bit of an idea of what a billion is. When we start looking at trillion, that gets really tough. When you start looking at a trillion, you're looking at a pretty big number, aren't you? And we'll be looking at those numbers next week. Thanks for your attention.